Building experiments within the Synthace user interface is easy, flexible, and user-friendly. Within our workflow builder, we can use elements as building blocks for each of the experiments that we're doing. Elements are easily chosen from a library, and each of them contains a set of parameters that define what that element is encoding. In this case, we're defining a set of liquids, such as standards and buffer, and we can then do a dilution of these standards according to parameters that we can define. Parameters such as concentration of different components in SI measurements that are known to scientists, the number of dilutions, as well as the plate, name and type in which this dilution will be performed. Information is easily passed between elements by connecting them between them, so elements are automatically customizable. We can keep adding new elements from this library in order to expand this protocol. In this case, we're adding further liquids such as samples and defining the plate layout for these samples, aliquoting them down the line. In this way, we are expanding our assay from a dilution and aliquoting further into more aliquoting steps and what will eventually become a Bradford assay. Thanks to this flexibility and transfer of information between elements that can be easily modified, we can transform assays like these from Bradford assays into picogreen DNA quantification assays, for example. The builder interface is very easy to use. Zooming in and out for best view is always available to the user. Here we're completing the aliquoting of our samples and standards and defining the Bradford reagent for the end. In the same way as we've defined liquids previously, we can use the same element to now define this extra Bradford reagent that is coming into this assay. Ultimately, we can then tell the Synthace platform that we would like to mix this Bradford assay onto the samples and standards, and we have a complete full Bradford assay workflow. This workflow is now ready for simulation and execution.